Not live yet. Assalamu alaikum,
Sure.
everyone. Thank you so much for joining in. It's good to see you all here. Sir Dr. Taimur Shakoori, he has very graciously offered his time and expertise to us, and I'm very glad and very thankful that he was able to join us and help us out here. Thank you so much for coming in, sir. And
no worries. The pleasure is all mine. And assalamu alaikum to everybody as well. I hope that this session will be productive.
So, sir will be talking about O levels and IGCSE bio. He will initially talk about what pitfalls are, how to attempt the paper, what are the requirements, what are topics that are recently added to the syllabus if there's something removed. And then obviously the floor will be open for questions, and sir will inshallah help you out with all of that. So with that, I won't get in the way. Sir, take it away, please. Thank you so much.
Thank you very much, Eamon. Thank you so much for having me in. Acha, assalamu alaikum everybody. For those who do know me, my name is Taimur Shakoori. So I've been teaching biology at the ONA levels for the past decade or so. And I thought that, what better way to actually have an impact and, have a discussion with everybody concerned, what better than having a live session for this purpose. So I'm going to also take a look here and see if I can also see any questions that you guys might be putting into me. So I'm just going to open that link too as well. Just bear with me while I do that. And yeah. Okay, so I hope I'm audible as well as everybody can actually hear what I'm saying. I'm just going to, if you don't mind, shift my internet so as to ensure that there is no issue with it. Okay, everybody, I hope you guys can listen in. And I hope that there's no internet issues because I've just shifted my net. Either way, so what is the idea? The idea now is basically to check, first of all, because we're dealing with both 0610 as well as 5090. So I'll be looking at your questions alongside as well. But just before we do that, I'm sharing the screen in front of all of you. My idea is to first discuss what are the things that are relevant. Now, hopefully within the 2 to 3 years that you all have been studying for this whole, for this time, hopefully you do know what is the requirement as far as the, syllabus is concerned. So all I'm gonna do is first, I just need you guys to know whether or not we understand what is part of the syllabus and what is not anymore. So I'm going to first go with the things which are part of the syllabus of 5090 now, because understandably so that 5090 actually experienced the most changes as far as the syllabus is concerned. Now, this particular document, I'm just going to use the help of this document, understanding that how it went, how to go about it. So for example, the the topic of classification for those who were involved with 5090 was never part of the syllabus, just like it was in 0610. That's the IGCSE one. So classification ka chapter pura aa gaya. So that means that your division of the syllabi into the 5 kingdoms. So basically that means kingdom Animalia. That also means kingdom Plantae, whether it's prokaryotes, which includes the bacteria, or if it talks about fungi, which are, of course, the 4th separate kingdom. And if so, you want to add that as the 5th one, that's viruses. And finally, there is another one, which is protoctist. So anyhow, this is basically the different kingdoms. And then when they're never there before. So the structures, rather now, viruses, bacteria, and fungi, which used to be part of the chapter called microorganisms and biotechnology, which is not a separate chapter anymore, they're included in this. And the important thing I would actually point out here in viruses, especially, that a lot of times what kids do is that they tend to forget what the name of the outer layer is. So if it's a layer that seems to have these circular spherical structures, remember that these individual circular spherical structures are called capsomeres. And overall, this whole structure, which consists of lots of capsomeres, is known as the capsid layer, otherwise known as the protein layer. So it is not a cell membrane, rather it is a capsid layer or a protein layer. And that's typically what every virus will have, irrespective of whatever. And in the middle, there will be a nucleic acid core, and the nucleic acid core will either be an RNA or it will be a DNA. Sometimes kids mix up and think that, okay, it'll be both of them. So there'll be only the RNA or the DNA. Now, why is that relevant? Is because although you have a P2 and P4 tomorrow, respectively, but, and when I say P4, I mean the P4 of IGCSE. So I hope the 5090 ones don't get confused that I'm talking about ATP, but I'm not. So it's only the P2 for them and the P4 for IGCSE. So point is that the RNA or DNA, it's got to be one of them. So for example, sometimes in an MCQ question, they ask you that what are the possible things that a virus can or cannot have? And they sometimes say that it doesn't have an RNA. So that can be the scenario, because if it doesn't have an RNA, then obviously what's the other thing left? Obviously it can either have a DNA instead, right? And in some scenarios, it can even itself, this capsid can even itself be covered by another layer, which I'm making in red. And then it's got these fancy looking receptor proteins coming out. So you can call them, in the age of COVID, you call them spike proteins, which is a general term used. But from a CIE perspective, O-level perspective, you basically are going to refer to them possibly as glycoproteins, which is the best thing you can write, or you can refer to them as antigens, as simple as that, which we know are the foreign substances, or rather on the surfaces of the foreign substances that obviously attract the secretion of antibodies. And this whole thing, which is to which the spike proteins are attached specifically, they're often obviously referred to, this is going to be the fatty layer. So this is going to be the fatty layer. So this is the reason, by the way, why during COVID you were washing your hands 20 seconds with soap, because soap gets rid of the fat. So anyhow, this was important to remember that viruses have this. The important thing regarding bacteria and fungi, also remember from an exam point of view, is the bacterial cell wall. So we talk about cell wall, and we know that the cell wall of the plants is made of what? It's made of cellulose. But the cell wall of bacteria are made of peptidoglycan. That's the term you use. Or the other term, if you're going to use, the other term you will use for this is murine. So that's interchangeably used. So either it's peptidoglycan or murine, and that's talking about the bacterial cell. As far as the fungus is concerned, of course, their cell wall is made up of what? Their soluble small made of the structure called chitin, which is also what the exoskeleton of arthropods made of. So please make sure that you are able to differentiate rather between the cell walls of both. So anyhow, all of this process and these structures and everything related to it is going to be part of the classification chapter. Of course, the implications of fungi and the bacteria, etc., they're also going to be put into process, but that is obviously the job for biotechnology and genetic modification. That chapter comes later. And you notice that all of them more or less the same. So I'm just going to really quickly skip over to the things that are no longer part of the syllabus so that you know for sure. And then obviously the new topics that are included. So the concept of malnutrition is no longer really asked anymore. Causes of tooth decay are not there anymore. Now, Doesn't mean that you shouldn't know which teeth there are there, which you know that they're incisors, canines, premolars, and molars, ICPM. And of course, that's basically what you have. And of course, that they are present in this manner in each jaw, which means that there are 32 in total if you're talking about the adult teeth. But you should need to know this stuff. So cause of tooth decay, of course, probably not that important. But the location of the teeth, right? Considering that the incisors come first, then the canines, then the premolars and the molars on each side of your jaw respectively. So that's how you form the jaw. So that's basically there. And you know, so that hasn't changed. The individual structure of the tooth is also preferably supposed to be remembered, which includes, of course, the enamel, E for enamel, D for dentine. And of course, then you have the pulp. So that's over there as well. And that's basically it. Kidney dialysis was out of the IGCSE as well, and it's out of currently the O-level syllabus as well. The reason for that being, of course, is that they've tried to streamline both the syllabi in accordance to the requirements of A-levels, as you know. Structures of the brain no longer required. However, as a sidestep, I will still say that when you're looking at the brain, and fine, agreed that you don't necessarily have to remember anything related to the structure, but I would still say from the perspective of the endocrine system or the concept of hormones, remember that this P-shaped structure that I'm coloring green right now, that's basically the pituitary gland, and that has a huge role to play when it comes to, you know, when it comes to basically hormones. So the majority of the hormones released by the body, whether it's FSH and LH, which of course are the reproductive hormones, as you know, or it could be ADH, which is basically antidiuretic hormone, which basically means that it is there for water retention, right? So, and of course that is controlling the osmoregulation. And next to it is a structure which is quite next to it. So that's the other green now that I'm making, and that's the hypothalamus. So possibly at least these structures know, if not their location, at least know what they're all about. So hypothalamus, remember, is said to be the thermostat of the body, which means that it's supposed to control the temperature, right? So this, until this point, at least, I think homeostasis se taluk rakhta hai hypothalamus. So obviously this is something that you have to know. Advantages of breast milk over bottle milk is once again out of there. Birth control, which means the, you know, all the substructures, you know, all the things which are related to, you know, to it is gone. Parasitism, support movement and locomotion was a whole chapter. So that's out of the equation as well. Remember that I'm dealing with 5090 at the moment. What are the ones that are included now in 5090? The one of disease. And when we say disease, we're actually specifically referring to the infectious diseases, which means disease that are transmissible. In other words, they can be transferred from one organism to another through different vectors. Okay, so malaria, etc. So all of them are there. And in fact, you should know the names. So cholera, which is written below as well, is part of the syllabus. So is malaria. That's part of the syllabus, which has a vector, which is the mosquito, the female Anopheles mosquito. And the pathogen is the plasmodium for that. Then you have HIV, the effect of which is possibly not part of the syllabus as such, but it is still part of your syllabus in terms of how to control HIV, right? So that's over there. Importance of clean water, of course, it's important. Why are we referring it close to cholera is because we know that cholera is transferred through contaminated water and food. So basically it's through contaminated water and food. And when we say contaminated water and food, what do we mean? We basically mean that it is spread through the feces. So you cannot expect a person to sneeze cholera into you, but you definitely can expect that if you don't wash your hands, or if you know, you eat without washing your hands, for example, it can actually transfer. Coordination response in plants, this is the job of talking about tropism. So phototropism is the, you know, basically is the growth towards the, and based on the direction of the light. Positive phototropism will be based on the direction, you know, towards the light. Negative phototropism is the growth away from the light. And similarly, geotropism or gravitropism comes into play as well, where, you know, the roots grow downwards to the bottom, which means that it is according to the force of gravity. So that's positive geotropism. If it goes up, then of course there is negative geotropism at work, or negative gravitropism at work. In this sense, the main important hormone therefore would be a growth hormone, and that is known as auxin. So the importance of auxin will be necessary. Remember that as far as auxin is concerned, the important thing will be to ascertain that, let's say that this is the tip of the root or the shoot. And you know that this is the area which we call the meristem. And meristem, remember, is all those areas of the plant which are actively growing. So that's the meristem. And in this meristem, what you have is that, of course, that when the hormones are, accumulated in one end. Okay. So what generally happens is that when they're accumulated in one end, they generally are going to diffuse to the other end. And once they diffuse to the other end, what does that cause? When they diffuse to the other end, just making it above, what that causes is that the movement of now the plant will be in the opposite direction, or rather in the direction, from where light, for example, is coming from. So it begins at one end, it diffuses to the other end, and then, of course, when it actually grows, it grows towards the opposite end. So this is one of it in a nutshell. Then we've got conservation of fish stocks. Now, this is really important, I would say. And a quick word on the conservation, especially process, is because it's become newly into the syllabus, especially for 5090, even if it did exist before. And so obviously this is for both 0610 and 5090. So I just want to basically point out here, and I do believe that this is one of the important questions that might pop up. You never know. And so I'm just going to make the use here of my notes, and I'm just going to actually make them up. So if you just bear with me here, so I can talk about here conservation. And within conservation, of course, there's a lot of aspects of conservation that are added to the syllabus. So we're basically referring here to fish stocking. And for that purpose, it's important to know that how this is supposed to happen, and what are the ways indeed in which conservation of fish stocks specifically can happen. So the conservation of fish stocks are what basically needs to happen is, first of all, number one, there needs to be some education, and that is a very generic term to use, I know, but you have to be careful, because in P4 or P2 for that matter, you have to be really specific in terms of answering a question. And by the way, by the end of discussing the overall syllabi and the changes, I'm obviously going to talk about the basic details towards the end of how to go about answering questions. So, we know that in certain parts of the world, the local fishermen overexploit fish stocks. So this is basically to prevent over exploitation of the fish stocks that are available. Okay. So that's basically it. And in certain parts of the world, remember that fishermen, what they do is they use a combination, unfortunately, of dynamite. So D-Y-N-A-M-I-T-E, dynamite, and the poison called cyanide. And what they generally do is they basically kill off basically they destroy habitats to kill off the rest. And obviously these methods kill all the organisms in the area. In fact, coral reefs generally get destroyed as a result of it. The coral reefs are very potent habitats for the ones on the aquatic region. So a great education can help them to actually prevent it, because habitats should not be destroyed for this case. And, that's basically for education. Then to ensure that if there is a lack of fish around, then just ensure that there are seasons where there is no fishing at all. Okay, so close season, obviously, is that part of the year when it's illegal to fish. And you could do that, from the perspective of the government policies. So your questions in P2/P4, whatever your variant is basically to also look at that. What is the examiner asking you? That, for example, are they asking you how the government will impose this scenario of conservation, or how scientists, for example, will do that? So close season is something that the scientists obviously will not be able to do. It is something that is the job of the government, or maybe the NGOs, etc. Education is something that both the government as well as the individual research organizations can do. Number three is then you can provide them protected areas. For protected areas, obviously, we could talk about, for example, the marine protected areas, right? And in terms of marine protected areas, we know that we have to basically give them a specific geographical space to either any of the species or a specific species. So we can enforce this, that, achieve long-term conservation. So obviously we need to find a way of preventing the capture of these fish. So, the fish stocks can continuously rebuild. And this is basically one of the examples. This is fisheries. And we want the fisheries to somehow become sustainable in a way that it doesn't, go to waste. Then the other thing, which is basically to control, net types and mesh size. So the types of nets that is used, as well as the size of the mesh. Again, I'm speaking about this particular part of the syllabus right now because I believe it's some part that often gets missed out. And so fishing net types, ka matlab kya hai ki, for example, you cannot use trawl nets. So trawl nets cannot be used. So trawl nets can be very destructive. And obviously trawl nets इस्तेमाल करके होता है कि coral reefs का भी तबाह हो चुकी होती हैं. So, and trawl nets need to be banned, of course, in some parts of the world. And that obviously protects the marine habits. So that needs to be removed. And of course, then you obviously need to allow the mesh size to be such that it should be large enough to allow young fish, the young fish to escape. You don't want to kill the young fish. Take it. And even for the large fish, and this is to prevent the trapping of the gills of the large fish, because unfortunately that can happen as well. So not only you're not able to catch those fish, you're obviously allowing the fish's gills to be captured in the mesh when the fish is trying to escape the net. So obviously, so that's not going to happen. So again, what scientists generally have done is they used LED lights on top of these fish nets or gill nets. What's the purpose of the LED lights? So obviously at night it can be easily used. So, it can reduce the accidental catching, for example, of other organisms such as turtles and dolphins. And of course, they don't reduce the amount of amount of fish that are caught, but eventually they can, prevent high bycatch rates. So the idea is to reduce high bycatch rates. Bycatch rates generally mean that it accidentally catch things that you do not need. Okay, so this is a good welcome addition. Number 5 will be to, again, discuss some legal quotas. Legal quotas ka matlab ye hai ki, for example, in Europe, they've got this policy, in which they can manage the fish stock and protect the species. So again, you can allow the fish to reach a breeding age, okay, so that's to maintain their population. And quotas are sometimes not popular because fishermen really don't, listen to them. They catch way too many fish. And to hota ye ke even if they catch extra fish, so for example, they get caught, if they do get caught, and then they're not allowed to sell them. So the surplus fish caught by fishermen are taken from them and not cooked, of course, but they're thrown back before they're dead. So that's basically what we do. And finally, of course, you've got monitoring scenarios. You need teams out there, whether it's from the government or ecologists. So the fisheries, basically, they're fisheries inspectors, and they monitor commercial fish that are caught. They record the catches, and of course, then they also make sure that the fishing boats are licensed and all of this. So please go through this, especially if you're looking at the new book over here, which I'm going to share with you as well. Of course, I'm not asking you now to change the book altogether, but in case you are using the new book for CIE, it's for 5090 for 2023, that's something you need to look at. Another addition thing that I basically wanted to talk about, and there's no, telling what kind of questions might come. And this has to do with conservation in general, but fisheries is specific. So a very important example, I would say, is the example of the tomato fish project. Okay, this was initiated by Germany. So that's what they've done. So the idea is to grow tomato and fish together. Tomato and fish together. Now, why together? Because the idea is the advantage, rather. So the reason why we are doing this together, we are growing tomato and fish together, is because it turns out that even though they're both completely different organisms, they are completely different kingdoms altogether, but they both need the same environment, a very similar environment to thrive. For example, the temperatures that both need ideally is approximately around 27 degrees Celsius. You give the tomatoes and fish, and when I fish, by the way, when I talk about the fish, of course, I'm obviously not talking about every fish. There is a specific fish. And if you can remember the name, great, but it doesn't need to be important that you remember it. So Tapelia is the name of the fish, and they're basically omnivores, which means that they don't necessarily have to, be given some carnivorous meal to eat all the time. They can easily just take it from the plants that are used to grow the tomatoes. So that's basically the reason. Now, where is this all happening? It is basically a makeshift greenhouse, right? And it is obviously, we generally call, the word we use for it is aquaponics. Now, aquaponics is obviously derived from two terms, which are aquaculture. And the second term, of course, is hydroponics. Now, what does it mean? So aquaculture basically, as you know, is farming inside the water. So using the water to basically breed something. So fish farms, for example, are aquaculture. Aquaponics are hydroponics, basically. Hydroponics basically is to not have soil, but rather it is to have a water-based nutrient solution in which the plants are able to grow. So this is basically a soil-free growth and water-based growth medium. And what are the advantages and why do we do this? Why are we doing this? Why are they growing them together? So the best thing about these, as the topic suggests anyway, is to conserve resources, right? Because it's a cycle in itself. So for example, fish apparently give off less carbon dioxide during their cellular respiration. The reason for that, of course, one of the reasons is that they're cold-blooded. So that means that lesser carbon dioxide into the atmosphere, and we know that when there is excess carbon dioxide, what happens? And generally, we don't need to put in too much food because they're omnivore, they can even eat the plant. So lesser carbon dioxide here, lesser carbon dioxide, there is lesser food, and water needed as well because it's happening within the water. And we only use a little bit of solar energy, which once again is renewable energy. So that means that this is not absolutely, but more or less emission-free. It is more or less emission-free. And it's also healthy as well, no doubt, because we're talking about fish here. Tomatoes, everybody knows how good they are. But for example, fish, you get omega-3 fatty acids from the fish, right? Your chapter of biological molecules, which means that they are unsaturated fats. And remember, unsaturated fats are supposed to be the good fats. They're the ones which have more than one scenario where they have carbon double bonds. So this scenario here and this scenario here within the fatty acid chain is that. And that is good because that allows easy hydrolysis and easy digestion of the fat. Okay. There is no waste overall because any waste that is created is obviously taken up. So for example, we know that fish give off ammonia, which is of course a waste gas. Now the good thing is the bacteria, or around, can actually undergo what? They can undergo nitrification, right? They can undergo nitrification, convert it to nitrites, and finally into nitrates, right? So the names, if you want to know about the two nitrifying bacteria, one is called Nitrosomonas, which is supposed to be there, and the other one is the Nitrobacter. So if you want to remember these names, you can as well. So again, nitrates is something that the plants can easily use. It can easily be used by the plants by the process of active transport, right? Ammonia, probably not that easy to do. In fact, probably not at all. So that's basically it. And of course, last but not the least, it is a closed loop system. So what is meant by closed loop system is that it basically is going to be useful for those areas which are too dry. So those areas which are too dry and don't have enough water in them, that means that the water can keep, circulating. Now, what does it mean? It means that, for example, if you've got the fish and this is the cycle, and you got the tomato, for example, this is the tomato over here. So generally, we know that the fish is offering water because that's where they are. So tomato is getting the water. The leaves rather are undergoing transpiration, which we know is the loss of water vapor. And that is then trapped in something called a condensation trap. And from the condensation trap, that water can easily now go to the fish, so to speak. Okay. Now, it is obviously during this time that, the tomato can actually make use of the water to even photosynthesize. And of course, Any carbon dioxide that the fish release can go into the tomato plant for photosynthesis, and any oxygen which is released as a byproduct can be going into the fish as well. So it's a win-win situation, and there are many things that are useful for it. So again, I'm not necessarily saying that you'll get a question asking you specifically regarding that how do necessarily tomato fish project works, but it could be part of the question. You never know. And more specifically to 5090, because I'm not exactly sure how much you probably will have done this within class. But obviously, whatever I'm talking about is relevant to both, by the way. So this is basically in a nutshell of what I wanted to talk about when it comes to 5090. All right. So this is basically the changes there. This is 5090 alone. Speaking about the paper, obviously pattern, as everybody should know, that paper 2 is the name given to it for 5090, right? And you know that it is 1 hour and 45 minutes. So 1 hour, 45 minutes. So that's 105 minutes. You have a total of 8 questions, you guys, take it. And of course, and each question, of course, is going to be about 10 marks each. So that basically means about 80 marks. And remember that this time around, there will not be any choice. There will be no choice. Just like in previous times, there used to be a section A, B, and C. So section C used to be answer 1 question out of the 2. So no choice now. Either way, it doesn't really matter, because at the end of the day, it won't get an A-levels. And secondly, the choice did not necessarily mean that aap kuch hissa syllabus ka waise hi chhod sakte the. So it's not something that's out of the blue, or not something that's necessarily a disadvantage, because think about it this way: you don't have to waste time in reading an extra question to first decide in the next 3 to 4 minutes that which question to do. So don't worry about it, and just go with the flow. So 1 hour 45 minutes for you guys. I would probably say that 105 minutes are there. And that means that you have 8 questions. So if I do the absolute math of it, of course, then it's most likely going to fall to a scenario where you're going to use about approximately 13 minutes per question. But I don't want you guys to do that. I want you to think that, okay, we have a maximum, I would say 96 minutes, maybe 96 minutes, kar lo behtar hai, because that will give you 12 minutes per question. And that will also provide you an extra 9 minutes. Now, I'm being really mathematical here. So of course, don't need to be that mathematical. So maybe an extra 7 to 9 minutes just to go through the question. All right. And of course, I'll talk about how we go through the questions in a bit. Before we do that, I'd like to actually quickly address the 0610 team to see whether or not they also have understood what the syllabus changes have been. So for them, the syllabus changes are not very vast, but again, I'll simply go through it. What are the syllabus changes and the definition? So first things first, the term definition apparently for 0610 has been replaced with describe. Now that opens up a new ball game. Definition used to mean definition. Describe का अब word अगर ये replace कर रहा है definition को, तो उसका फायदा भी हो सकता है, नुकसान भी हो सकता है. फायदा कैसे है कि description का मतलब ये है कि you don't necessarily have to use those exact words, which obviously you don't have to, but you're obviously looking for biological terminology, but probably you're not looking for कि भाई ये मैंने definition CI के मुताबिक IGCSE के मुताबिक नहीं लिखी तो won't get the mark. You can basically describe it by using examples as well. So for example, definition of gene, right? And let's say definition of gene was for 3 marks, and let's say that I would say definition of gene that it is a section of DNA, right? Or a portion of DNA represented by a specific sequence of nucleotides, right? Okay, section DNA represented by a specific sequence of nucleotides used to code for producing a particular, you can say protein or polypeptide, right? You can say this is a unit of inheritance. And you can say that it easily transmissible, well, not transmissible, but it can be inherited, from parent to offspring. And yeah, so that basically is it. So if you're looking at this definition, or you're looking at the terms which are important, you probably think that, okay, everything is required now. But the good thing about describe is, I'm not saying that you should not write all of these points down. All I'm saying is that let's say you wrote that there's a section of DNA that produces a particular protein. Yeah, you're not writing specific sequence of nucleotides, for example. Then the good thing is that you can even write an example. That example, for example, the hair color or hair itself, nails, finger. Sorry, not say nails. Or sorry, like what? The shapes of your hands, etc. Whatever. All of that comes in the category of genes, which represent a particular trait. So again, the definition question is now replaced with describe. Characteristics of living organisms. Again, if you recall in IGCSE, most of you are giving a P4, but there's obviously a P3 as well, which means that there's a basic core one. So they're just basically talking about how some of the material that used to be in core now goes into the advanced and so on and so forth. So the section on cholera has been, for example, moved into the supplement, and the section of skin is also moved from there to there. And then it's important to notice about the use of the AI and the IVF. And I need the guys who are in 5090, the girls and boys, in 5090 to also have the ears up, because even this could well be part of your exam tomorrow as well. Even though, it's been added to the supplement as far as the IGCSE is concerned, but for O levels, it's a new thing. So AI stands for, by the way, artificial insemination. And as the name suggests, it basically is the process in which the male gamete is somehow inserted, and the pregnancy is caused. So artificial IVF basically is in vitro fertilization. So we're just going to talk about that in particular, in case some of you are non-cognizant of it. Okay, so we're going to really quickly talk about the concept of conservation through these methods, which are obviously the scientific methods. So we're going to begin with artificial insemination. So just a very quick word on it, in case it comes out in any way. So I'm talking artificial insemination. And then, of course, we're going to deal with IVF very quickly. Okay, so these are what kind of programs? We're giving them the name captive breeding program. So keeping the organisms which are apparently in danger of either endangerment, of course, or in danger of extinction. So captive breeding programs are techniques. So the idea regarding this is, there is reluctance, because not every organism is going to be comfortable in an artificial scenario in a national park or probably in a zoo. So reluctance to mate is one of the reasons why we have to opt for these things. Take it, and that means why that we'll simply collect the sperm from the male. All right. So the collection of sperms from the male animals, and then we have to artificially introduce. We have to artificially introduce them into a female's reproductive system. Okay, that's the idea behind it specifically. Yani, and of course, for that purpose, now, what will that do? Of course, we hope that will cause fertilization of the egg, right? And of course, hopefully, a viable embryo is made. I'm using a word here called viable. Viable means an embryo that can survive. An embryo that can survive. Can survive means it will survive. It will go through the pregnancy, and of course, it will grow up and become what it is. The organism is. Now, the sperm can be used either immediately. So it can be used immediately, writing it above, or it can, generally be, it can be frozen and stored. So frozen means it can be stored for a little period of time, after which it can be used whenever, the time is right. Maybe the female is not yet ready, or vice versa. So this obviously increases the genetic diversity of the species. That's the reason why we do this. Genetic diversity of species is increased, right? We are probably increasing what we call the gene pool, which basically means the same thing, which means that there is more, I'm going to use a word here, there is more heterozygosity. Heterozygosity ka matlab hai, there's a chance that there's both possible alleles of that particular gene. We know that what alleles are. Alleles are different forms of the same gene, that both alleles can possibly be part of it. And that's good in what sense? That's good in the sense that, if there's any kind of disease or any kind of, disease related to homozygous recessive or homozygous dominant, then in heterozygous scenarios, you can afford to basically be done with it. Then we've got, what else do we have? So yeah, so this basically has been successfully used in Saudi Arabia. Obviously, you don't need to know necessarily examples, and it's done in that sense. This is basically captive breeding program number 1, artificial insemination. Captive breeding program number 2 is IVF, also known as in vitro fertilization. Okay, so that's number 2, and that obviously, literally, IVF means in glass. Obviously, we're not going to use simple glass. We're going to use proper labware, so most likely in test tubes. So that means that the fertilization is supposed to take place in vitro. In vitro means, sorry about the spelling of fertilization, is single L. It's supposed to take place in lab glassware. Quite literally, you have the sperm as well as the egg cells, and you mix them up. And that's where the concept of test tube babies have come from. See, okay? Whether it's humans or others. So sometimes, once again, why is this happening? Why do we need to do it? So in the first scenario, remember the mating is not happening. And sometimes females, the condition which happening is that females at times simply are not able to breed naturally. It so happens that their placenta or their amniotic sac or something of the other, the pregnancy is getting failed repeatedly. And we can't afford that to happen because we don't have too many females left of that particular species. So we're hoping that we can get some more organism out of it. So the idea is to produce viable eggs. So again, similarly, it could even be a case that males are not producing viable sperm. So maybe there's a problem with the males. So either way, whether it's a problem with both, which again is a problem, but then if it's the same. So the idea is that the females can be given fertility drugs for this purpose. So we give them fertility drugs, again, which causes her eggs, lots of eggs release. So releases a lot of eggs, which means what process am I talking about? I'm talking about ovulation. So it releases a lot of eggs at the same time. And then, of course, they can be collected. And then obviously in a lab setting, the eggs quite literally are mixed with a solution which has sperm cells. And obviously the seminal fluid is done. And, after that, hopefully whatever fertilization is successful, they are basically inserted, into, through the cervix, which is the neck of the uterus, into the female's body. And that basically has been successful for rhinos and for tigers as well over the years. Provided that, the eggs can be frozen as well. It can be stored for long periods of time. In this case, so can the sperm. So this is basically one of the 2 things as well that I wanted to point out, and which is part of the syllabus generally for 0610. Okay, topics in 0610 no longer part of the syllabus. Just like in 5090, you don't have kidney dialysis anymore, as you know. The voluntary and involuntary actions are not that important anymore. Yeah, reflex action is something that you should still know, which is an instant involuntary response to a stimulus. Instant involuntary response to a stimulus. So that's something that you should know. You should also continue to know that reflex action is an action, but reflex arc is the route taken to carry out that action, right? From the receptor to the sensory neuron, to the relay neuron, to the motor neuron, and so on and so forth. Explanation of why antibiotics kill bacteria, not viruses, apparently not useful anymore. Section on misused drugs, not useful anymore either. So we're basically talking heroin, and etc. are not important for that purpose, and because they're not there in A levels anymore either. Having said that, though, we probably will still want to continue to know about nicotine and remember what the impact of nicotine is for smoking, because that is still part of 5090, and it should be part of 0610 still as well. Labor and birth, advantages of breastfeeding, all out of the window now. And apparently roles of hormones in controlling the menstrual cycle apparently is still not there either. And of course, how HIV affects the immune system. This is not there for 0610 anymore for sure. However, for 5090 people, still remember the HIV is supposed to affect what? It is supposed to affect the particular T cells. T cells are specific lymphocytes. Remember, lymphocytes are the antibody producing cells, and they are on the surface of these T cells, certain receptors called CD4 receptors. And it is the CD4 receptors to which your virus HIV that is, tends to attach itself. So again, that's one of the aspects of it. And the CD4 receptors then allow the HIV to enter. And then, of course, that HIV replicates and then it continues to attack T lymphocytes. That's one of the aspects, and one of the diagnoses of HIV is to look at the fact that आपके blood test में T cells जरूरत से कम होते हैं. ठीक है? Inheritance of sickle cell anemia ain't there. Definitions of fitness and evolution not there either, and so on and so forth. Nuclear fallout and acid rain. Apparently, the details not there anymore. Somebody was asking the other day as well. However, a little bit, there's no harm in knowing about acid rain. That it's simply merely it's simply the water in the atmosphere combining with either the with sulfur dioxide, or it's the water combining with the oxides of nitrogen. Either way, you're going to get sulfuric acid, or you're going to get oxides of nitrogen. So any ammonium or something of that sort. Take it. So this is basically in a nutshell what it's all about as far as the syllabus coverage is concerned. Now, I just need to be looking at and check if we have So basically, if you give me a sec, I'm just going to actually share with the screen now for a little bit. And we're looking at here. Okay, everybody. So this is basically what the one was there. Okay. I think I'm lagging a bit. I really hope that everybody can listen to what I'm saying. Yeah. Right, guys. Any questions before I begin with any particular topics or questions? My idea is to talk about the March 2023 0610 question paper, which once again, please note, does not mean at all that 5090 needs to go away. It's very relevant because, they kind of streamline things a bit. And yeah, so still looking for questions. No questions. Okay, I'm going to begin. So let's not waste time. We almost have an hour up. We need to almost halfway there. So I'm going to restart my screen share. And we are going with a few questions coming in. So I'm going to use basically two papers today, if possible. One is going to be a paper from 0610 and possibly one from 5090. So I hope the screen is shared now. Alrighty. So if you look carefully at this, alrighty, so again, there's no point of doing each and every question. For 0610, again, once again, your time, of course, as you know, is much lesser than the 5090 people, right? However, having said that, does not mean in any way that yours is any less challenging. So we empathize with you. However, having said that, though, the number of questions will be about slightly lesser, right? It's about slightly 6 questions, although the number of marks is not that much of a difference. So your work cut out for you guys, of course, you've got 75 minutes, all right? And for you guys, I would probably say that, once again, depending on the number of questions, I would still say that at least reserve the last 6 to 7 minutes for the sake of, revising. While you're doing a question, please make sure that you are aware. This is for both 500 0610, aware of the command words, absolutely essential, no worries whatsoever. Command words, and this is going to be doing well for you as far as both O levels and A levels is concerned. If it's describe and at explain, it could be state, it could be suggest, right? It could even be the following. It could even be compare. It could be evaluate. It could be comment. Now, I'm doing this from the top of my head of the ones that I've seen in previous papers, as well as of O and A levels. And of course, so basically some of the ones which are, I mean, obviously there will be more than that, but some of the ones that I actually was looking to make you understand that when it says and of course, the last one is outline. So when it says describe, then you are just talking about the what. For example, if there's a question that is giving you a graph and it's asking you to describe the graph. So for example, there's a sigmoid population curve. Once again, for the O-level people, please keep your eyes raised to this sigmoid population curve. So that curve actually assumes that with time or resources, the population of any given species, so for example, toads or frogs or rabbits, whatever, it begins to go up slowly and then goes up immediately, and then it basically levels off, and then finally it goes down. And what happens? It simply, goes down. So basically, that means that there are two aspects of it. There's A, then there's the aspect where actually is leapfrogging up. So that's B. Then there's another one which is supposed to mean that it's a bit stationary. So it's C. And then finally, it's going down. It's D. So A ka matlab kya hai? A ka matlab ye hai ki it's basically the the phase in which the growth is slow. So slow growth, right? B is exponential growth, right? Because doubling it is easy. But the fact is that after it doubles, then it comes to a scenario where it's the stationary phase, right? And after the stationary phase, we've got the dead phase. Now, please note that while I'm explaining this, I'm making a lot of things. I'm doing a lot of things which you are not supposed to do, and that is I'm not supposed to draw arrowheads. Please note, I am not supposed to draw arrowheads while labeling. The only time I can draw arrowheads is if I'm showing a direction, for example, in a food chain, or if I'm showing the direction that the next step is this. But if I'm labeling, I need to not draw the arrowheads. The only reason I'm doing it here right now, of course, is because it's because I just need to explain, and that's basically the reason why. So the first phase, which is slow phase, generally has a word. We call it the lag phase. You know that. And B is the one with exponential growth. It's a quick growth, so that's called the log phase. The C is the stationary phase. The name is given as it is. So, and the fourth one is the dead phase. The name is given as it is. So if they're asking you to describe it, you literally will describe that at what time is it slow till what? Then at what time does it exponentially grow? And what time does it become stationary? And from what time does it start to decrease? And what is the population? From what population to what population it does that? That's basically the description. No matter how many number of marks is, describe ka matlab ye hai. This is basically what it is. And then you've got the word explain. So sometimes the word describe and explain both are given, and sometimes it's only, describe or only explain. Okay. So the idea is that if it's explain, what does that mean? But that means that that's the why or the how behind it. So for example, if they're asking you to describe and explain this graph, then you can mention that initially it takes a while for organisms to start to grow based on the changing environmental situation. And then the scenario where you're talking about this area, which is the log phase, this area, which is the log phase, this is where you can say that this is the scenario where, because each time, the population is doubling because of the resources which are plenty, maybe the prey is plenty, so on and so forth. So that's basically, so this is basically what it is. And then of course you've got the one which is stationary. So for stationary, of course, you have a scenario where it's because of the lack of resources, it becomes consistent. And of course, then it all starts to drop. Because they start to die out. So anyhow, this was basically it. State ka jo word hai, uska kya matlab hai ki basically you just need to state whatever it is, and without any description or explanation, it's going to be simple. Outline is quite similar, which is the last one. And obviously, if it's a sequence of events, for example, the sequence of events that are supposed to report how much the how the nerve impulse is traveling from a finger that has been burnt to the effector, which eventually causes the hand to be raised from the source of the heat. So that sequence of events is important because that's the only time, by the way, jisme aap numbering kar sakte hain and not otherwise. Suggest is a word that you're going to use, of course, which may have more than one correct answer. Comment. For these three terms, whether it's IGCSE or O levels that is, please remember that you will give both sides of the argument and you won't just talk about one side of the argument. For example, if it's asking you about the advantages or the implications of genetic modification. By the way, the word is now genetic modification instead of genetic engineering. They put that word in there. So that's basically the idea. So both sides of the argument are in there. So genetic modification, what are the implications, for example? So implications could be advantages and disadvantages both. So if the number of marks given is 3, then it means that you need to at least add one disadvantage or one advantage apart from 2 disadvantages / advantages, right? So again, that's basically one of the reasons why you want to do that. And again, the number of marks will obviously determine, but at least one side of the coin. So evaluate me, comment me, compare me, ye dono cheezein aayengi. It's an upcoming command word. Please make sure that you guys are cognizant of this. Okay. So this is basically the scenario. Okay, so I'm going to now discuss this particular question now. So this is basically the 0610 paper, but we're going for this. It says, and I hope you guys can actually help me out on this, if possible, say state the letter that represents the blood vessels that contains blood with the highest concentration of oxygen. So we're looking at the one which has the highest concentration of oxygen. And of course, that should be what? That should rather be the one that comes out through the aorta, right? And through the aorta, that's the one that should be. So the highest concentration of oxygen is the one with the aorta. So your answer should be what? Anybody? So here's the idea. Where do you think is the highest oxygenation? Look at this. This is the blood vessel, the one that's coming from the B, which is known as, once again, what? The pulmonary, what? Vein, right? So pulmonary vein is the one that carries the most oxygen, because by the time the aorta is supposed to carry it, there's a good chance that the oxygen can go into the heart muscle as well. So that's a common mistake that can possibly be done. So please make sure that you guys remember that the pulmonary vein will have the highest possible concentration of oxygen, right? So your answer to first one is B, of course. Take it. Then the next one is state the letter from figure 1 that represents the hepatic portal vein. And of course, that's the vein that is going to be carrying the blood from the gut to the liver. Which one is that? Of course, that will be C. So obviously that is C. Then state all the letters from figure 1.1 that represents arteries. So we got to look at the arteries here. Remember that what are those blood vessels? These are the blood vessels that carry blood away from the heart, right? So the ones that are carrying blood away from the heart are what? They're the following. They're obviously, you've got this one. What is this called? This is called pulmonary artery or something else. This is called the aorta. Yeah, that's right. So aorta is obviously the first one. That's one. What is B? B the vein carrying blood back to the heart. So we don't consider that. What is E? What is E? So notice that E is carrying blood away from the heart. Once again, that's the pulmonary artery. So that will also be considered as an artery in this particular scenario. We look at C. C is a vein. We already said that. So that's not there either. We'll look at D. What is D? It came away from the heart. It's going towards the intestine. It's basically called the iliac artery, which is the artery that carries oxygenated blood to the intestines. So your answer to this is A, D, and E. Not B and C, of course. And then it's asking us to explain the importance of the part labeled Y. And if you look at Y, you'll see that the Y is basically what? The Y is referring to the septum. It is referring to the septum. And of course, that's basically what it does, is that it separates the left and the right side of the heart. And it says explain. So I can't really, stop myself and say that, okay, it simply separates it. I have to also go ahead and talk about the fact that the reason that it separates the left and right side of the heart is because by virtue of this, it is also separating, thereby separating the oxygenated blood from the deoxygenated blood. So where is the oxygenated blood? The oxygenated blood is going to be on what side? It's going to be on the left side from the right side of the heart. So that will be the right side from the deoxygenated blood. So that's basically another one. Then we got state the evidence from the figure that the diagram shows a double circulation method. Double circulation. Now, again, this is a side step I'm taking here. Obviously, I will answer this question, but here's a common mistake that a lot of kids do. Sometimes they don't ask you just for the double circulation. Sometimes they ask you to talk about the closed double circulation method or closed double circulation or what it means. So please, have a ear out for both of you guys. Closed means that it is happening within a cardiovascular system, meaning within blood vessels, a closed system, not open to the environment. Now, what is this? This is in opposition to an open system. Open system is basically those of the gills and scales. That's basically what it is. Meaning that basically how the fish are constantly, their gills are constantly in touch with the external environment. So that's not what's happening with mammals. It's a closed double circulation system. And of course, double circulation system in itself means what? That, it basically means the blood, as we know, passes through the heart twice within a single circulation. Within a single circulation, that's what's happening. So this is what's happening, guys. And so in this scenario, how will the diagram show us? Okay, how will the diagram show us? So you can notice that the diagram is showing us the arrows passing through not once, but at least twice. Okay, twice you see blood exiting the heart, if you're just specific for the heart, and twice you see blood entering the heart. So you notice these two, the ones I marked yellow, and then you see these two, the ones I'm marking, or maybe those, the red. So basically that is showing us that the blood is flowing through the heart twice. And also the fact that there are two separate blood circuits. So one blood circuit has to be, of course, the pulmonary circulation, which of course is represented here. And the other circulation is, of course, the systemic circulation, which of course is across the whole of it. So there's two civil circulations here. So that means we should have enough to gather the 2 marks that we need for this particular question. We got to state the evidence so we don't have to go for a long explanation. So we mainly say that blood is seen, or blood rather is directed towards and away from heart twice in the diagram. Okay, we have to personalize this answer. And the second thing is, of course, that there are, it shows 2 separate circuits. That is pulmonary and systemic. Please note the word is not systematic, it is systemic. Systematic likhoge to answer galat ho jayega. Yes, Abdullah, that is correct. Blood goes to the lungs and to the body, circulating twice around the heart. That is absolutely correct. Here, obviously the importance of the septum is going to be not mentioned because we don't have much of a thing with it. Here's looking at part B. Figure 4.2 shows the pressure of blood flowing through different blood vessels as it travels around the body. So looking at the pressures. Now here, often a difference is made. And before I begin with the question, ज़हन में रखें कि capillaries का pressure notice करें और venules का pressure notice करें. तो जो capillaries का जो pressure है वो कम होता है थोड़ा सा, ठीक है? किससे कम होता है? Artery से कम होता है. But venules and veins का pressure उससे भी कम होता है. So I hope you guys can make sense of that, because the blood has to go through the arteries, then the capillaries, then the venules, and So it makes sense that the venules and veins have the lower one. Okay, so anyhow, venules are narrow vessels that connect capillaries to veins. We know that already. Blood vessels X in figure 1.2 supply blood to skin to surface capillaries and have a role in maintaining a constant internal temperature. So obviously, agar venules have between capillaries and veins, and what is X? X is naturally going to be the arterioles. They're going to be the arterioles. Now, generally speaking, it is said that if you write the spelling in such a way that it is phonetically acceptable, then it will be acceptable. But I will still say, do not make the mistake of writing wrong spelling. I will still say that go for the correct spelling, because when it comes to one-word answers, you are never sure what the examiner would want from you, because it's supposed to sound correct. Okay. Anyhow, so that's basically X. Then explain. So now the command word is back to explaining the reasons for changes in pressure seen in the arteries. So changes in pressure seen in the arteries. So we know this, that obviously the pressure above in the arteries is quite high. And notice that it keeps going up and down, isn't it? It is a constant crest and a trough. Okay? And we don't have to describe it. So we don't have to say that, for example, that it begins at 10 and goes to 16 and so on and so forth, because the command word is not saying describe. So we're not going to waste our time in quoting the data, so to speak, unless the question was regarding description. So what we're looking at is that the blood, it's basically going to discuss that, there's basically these changes in the pressure, the up and down changes in the pressure. I'm going to write the answer here, actually, because this will make it easier. So the up and down changes in pressure, firstly, are due to what? Are obviously due to the muscular contractions, of the heart, right? So that's why it goes up and then comes down. So there's stretching and recoiling. And obviously these are more specifically those contractions which are the ventricles. And obviously the blood is being pumped through the arteries. Okay? So that's basically what's happening here. And obviously this is only the arteries, so the blood is being pumped through the arteries. And it so happens that the time at which, they're constricting, that's when the pressure is greater. So there's a lot of stretching and recoiling that is involved to prevent the bursting of the arteries at one end, and on the other hand, to maintain the temperature, or rather the pressure. Okay. So this is basically that. And then, of course, so this is basically it. And finally, the last but not the least, says using the information in figure 1.2. So now what do we have? We have a scenario where we are looking at the information. So we got to quote the data now. So even though it gives the answer of explanation, we have to use the information given from the figure alone and not from our own minds, and to, what the adaptations or structural adaptation of arteries and veins are. So we've already mentioned here that, because the pressure is high, and that can only happen when, there's the capacity to withstand the blood pressure. Okay, so kya hoga? What will the wall contain? The wall of the artery will contain smooth muscle, okay, which is the muscle that is supposed to be involuntary. And it will also have, for the sake of being able to withstand the pressure, will also have elastic tissue, right? And what will this allow? As I said, it will allow stretching and recoiling. That is what basically creates the pulse. Okay? Also, we know that high pressure maintenance can be taking place. That will happen by the narrow lumen. So narrower the lumen, the higher the pressure is going to be. And of course, that basically is what we have for arteries, right? And this is basically for arteries. For veins, of course, is going to be different. Obviously, it is low pressure. So we do not expect a lot of smooth muscle. So smooth muscle possibly will be lesser, right? So that's basically it. And of course, because it needs, low pressure, so it's going to be a larger lumen. And of course, there should rather be valves to prevent backflow of blood. And these valves, by the way, are semi-lunar valves. So they will prevent the backflow of blood. And this is caused by low pressure. So by the way, remember that you should be able to discern that what is a scenario in which you're able to see, that what is the direction in which the blood is supposed to go. Sorry about that. So the direction in which the blood is supposed to move. So as an example, if this is what I made, how do I know where the blood is going? This is a vein. So what is the direction? Direction A to B or B to A? Yes, Iman, that is correct. They do have valves present and they have a wide lumen as well. Righto. So so the direction will be, what do you guys think? Will it be A to B or B to A? I'm just asking in general. Abdullah, give me a letter, A to B or B to A. Yeah, Mystique Universe, that is correct. That is A to B. It is supposed to be that way because you're supposed to allow the blood to pass in this direction. And thereby, when it does, so in case it fails to go forward and go back to the heart. And by the reason why I'm asking is because that definitely means that the heart will be on this side, right? The side where B is, because that's what veins do. They carry the blood back to the heart. Yep. So so the blood, in case it doesn't go all the way back to the heart, so इस area के अंदर blood जमा हो जाता है. So किस area के अंदर जमा हो जाता है? So I'm talking about the area that I'm marking green. Okay, assuming that the veins will basically close up. So consider like a three-dimensional point of view. And of course, when that happens, remember that on the sides of the veins, there are present what? There are present skeletal muscles. Notice I'm using two types of words for muscles there. One I used for smooth muscles, which is what the walls of the veins can possibly be made of. And the other that I'm using is skeletal muscles, which are basically surrounding the veins. And they are the voluntary ones, which are supposed to squeeze, or rather they're supposed to contract, to squeeze the blood through in case the blood fails to return back to the heart efficiently in time due to lack of high blood pressure. Okay. So this was basically it. And then let's talk about, so what I'm doing is I'm going to do question 2. There's a question that I saw somewhere around here from people addressing it. And yeah, someone was asking regarding a synapse. So bear with me. I'm sidestepping here because what I basically wanted was everybody to feel that, it's kind of not just interactive, but it's of use. So I'm giving a short giveaway here for that what a synapse is going to be and what is happening in the synapse in terms of neurotransmission, because this is new for the O levels, specifically, if not the IGCSE 1. Abdullah Kashif, your question is, shouldn't we write skeletal muscle instead of smooth muscle for vein in the question before that? Well, we're talking about the muscle that the vein itself is made of. That itself is smooth muscle, because for that to be able to contract or relax is not in our control, right? We cannot cause our arteries to contract their smooth muscles. So that means that they're involuntary ones. Skeletal muscles are the muscles which are surrounding the veins. They are basically the muscles which are connected to the bone. Okay, if that makes sense. So we're not talking about the skeletal muscles that make up the vein. We're rather talking about the skeletal muscles that are surrounding the vein, not part of the vein. Okay. And so for neurotransmission, we just have a quick word here for synapse. Synapse cilia. We are just trying to ensure that we know that this is one end of a neuron. And then, of course, the other is the other end of the neuron. And assuming that the neuron nerve impulse travels down the way here, like so. Okay. Right. And So number one is that the impulse travels to the terminal end of the nerve fiber, also referred to as the axon. Okay? So I'm talking about that. And when that happens, of course, what this spurs on is the creation of certain vesicles. I'm going to call them synaptic vesicles. Okay. Carrying neurotransmitter. And a common neurotransmitter that we should know about is acetylcholine, also called ACH. So synaptic vesicles carrying neurotransmitter start moving to the, you can say, cell membrane, or basically to the end of the neuron. So what happens is number three, they move towards the end. So that's what's being shown as number three. So these vesicles fuse with the membrane. And I'm going to call the membrane name presynaptic membrane because synapse se pehle hai. Okay. Number four is the process that I'm going to call exocytosis. Is that within these vesicles was the neurotransmitter, which by the way is a chemical. So this chemical comes out in the form of these small structures. These chemicals that come out. So exocytosis, what's happened here, of neurotransmitter. So this is that. Okay. Into, and I'm going to call this the synaptic, or the synapse, which is actually what it is, the synapse. And as a result, these, so number five will be that these neurotransmitters will try to go towards the postsynaptic membrane, which is to the next neuron. And on the postsynaptic membrane are present already these small receptors. So what's happened in number four is that the neurotransmitter in synapse, or synaptic cleft, if you will, are diffused. So notice the first word I use in number four is exocytosis. Number five, I'm using the word diffused. Sorry, number five. So this is number four and this is number five. So neurotransmitter and synapse are diffused onto the next neuron by binding to the postsynaptic receptors, postsynaptic membrane receptors to be precise, to the postsynaptic membrane receptors. Right? Abdullah, your question is, don't the vesicles containing electrical impulses merge with the presynaptic membrane, then proceed to diffuse along the synaptic cleft binding with postsynaptic neuron receptors? That is precisely what I said. I don't know if I said anything different, right? So you're absolutely on the button. Huzaifa Gulzar, ACH is acetylcholine, the name of a common neurotransmitter. If you want to know the name, remember the name for neurotransmitter, just remember acetylcholine. Much else is not going to be asked of you, so don't worry about it. Adeen, I'm not a chemistry teacher. Well, I could be. I am basically a doctor of pharmacy, but I'm just taking biology, officially speaking, in the schools that I teach here. Okay, head finisher, I'm just coming to that. Neurotransmitter and synapse are diffused onto the next neuron, binding the postsynaptic membrane. And number six, of course, that the same nerve impulse, so we can say the nerve impulse is regenerated. And that's that. So this could even, by the way, this could even be a nerve impulse regenerated and travels through the next neuron, or this could even be, the end, it could be the effector. So it travels through the next neuron. And by the way, this postsynaptic membrane that I'm talking about, which is basically this one here, the postsynaptic membrane, this could even be referred to as the dendrite. So it could even Okay, sorry about that. I think I logged out. So I'm just getting back into it. So I hope, Abdullah, you heard what I said about axon and dendrite. Head finisher, what part 3 are we referring to here? Goofy, Unreal, whatever your name is. Yeah, acetylcholine. Look, again, it's basically this. It's the name of a neurotransmitter. I just basically told you a name in case you find it somewhere in the exam. You don't get all confused at what they're talking about. So basically a neurotransmitter, and it basically excites the nerve cell is what it is. Okay? So it's a neurotransmitter. It basically, sometimes it regulates blood pressure, if you will. But this is not the point where you're supposed to try to learn new information necessarily. All I need you to do is just don't be concerned and don't be, taken aback in case you get to hear those words. Yeah, so we're getting to that, and head finisher, I'm not exactly sure what exactly you're asking. You can explain part 3. Part 3 of what, by the way? Are we talking about the previous question that we were doing, 1B, 3? I'm not sure that is the case, but either way, I can go back to that and see what it's all about. So, yeah, and so I want to basically discuss this particular question is because I'm going back to 061042, and yeah. So that's basically what I'm doing. So I'm just going to do one more question. Then the next question that I'm going to go for is what Yashveen Sania just talked about. That's different types of pyramids. Dietary needs of pregnant women, again, probably not that much important anymore. However, it's not a bad idea that you just remember that what are the general things that are important in diet. And you can basically talk about it. I don't think that's a big thing. Okay, Abdullah, your question is, there are axon and dendrite cells with different structure or something for each neuron. Look, axon is the nerve fiber. Dendrite is simply the receptor on the other neuron. Of course, they look different. So for example, if because I'm on this page now, so let's say this is a motor neuron. And I'm drawing the length of the motor neuron, so to speak. This is the cell body. So the V stuff that I've made, so to speak, this stuff, which basically can also be called the postsynaptic membrane. These are basically the dendrites. So the postsynaptic membrane is that of the dendrites. This whole thing is the axon, or the nerve fiber, which, by the way, you should know is covered by this blue colored stuff, if you will, and they're called the myelin sheath. Okay, so that's basically it. Okay, I'm getting some questions there, so I'm just going to go very quickly over this question. Then we have a couple, so I'm just going to try to get through this. All right. So once again, student investigation, digestion of starch apparatus used. We got the dialysis tubing. Remember, the dialysis tubing is supposed to be semi-permeable. That is the main purpose of it to begin with. And there's distilled water outside, starch solution inside. But then again, the important thing would be here is to look at the second part, the test tube B, which has amylase apart from the starch. So any guesses what's going to happen? We obviously know the starch will be broken down into a simple sugar. In this case, of course, it will be maltose. So that's the idea. And of course, maltose possibly is small enough to be able to cross through the dialysis tubing into the distilled water. Dialysis tubing is used to represent the cell membrane. Dialysis tubing material allows small molecules to move across it, but not larger molecules. Test tubes A and B were set up in the water pad at 37 to 30 minutes. Liquid outside the dialysis tubing in test tubes in A was tested with Benedict solution and 0 minutes and after 30 minutes. Now, why did we use a Benedict solution? Because we're hoping that the starch will break down to maltose, and maltose can easily be detected by the Benedict solution. The original color for Benedict solution is blue, so that shall be as it is. If you notice, at 0 minutes, it stays blue. That is obvious, but after 30 minutes, test tube stays blue because, of course, it only has starch. And remember that starch cannot be detected by the Benedict solution, but red means that it contains maltose, which is why it turns red in the first place. And remember that the colors does not necessarily have to be red. It could be anything related to green, yellow, orange, and red. If you forget sometimes what are the colors that you use, remember that the rainbow color represents VIBGUYOR. After blue comes green, yellow, orange, and red. And that is the colors that you're supposed to see should there be any reducing sugars in the sample. G is for green, yellow, Y is for yellow, O is for orange, R is for red. Trace amounts will mean green, a little more would mean yellow, even more would mean orange. And of course, if you have lots of it, then you get red. Okay, so using the information in the table 2.1, explain the reasons for the difference in results for the test tubes. And of course, now it's asking us to use the information. So we'll have to quote once again from the data as well. And we have to talk about the reasons behind it. So obviously in test tube A, there was no amylase, right? So starch did not get broken down. So that's pretty clear. Whereas in test tube B, there was amylase. Starch did break down to form simpler sugar, also called maltose. And maltose is able to cross the dialysis tubing, which is acting like the cell membrane. Remember, cell membrane is controlling what enters or exits the cell from a region. And this is obviously happening through the process of diffusion. So again, I'm only writing points. I really hope that you understand that this is not how you really going to write your answers tomorrow. I'm just writing the points in there, right? So your best case scenario is for you to write in prose form. It's going to be in prose form. And basically that's what you're supposed to write. So anyhow, so that's why the Benedict solution turns red. So this is basically one aspect of the question. That of course, look at the names of the missing enzymes. So substrate for pepsin should be a protein. The product, of course, now, here's the thing, the product can be written as amino acids, for sure. That'll be correct. Or it can be written as peptides. Peptides basically means what? Peptides means that pepsin may totally break down Nikia protein. It has broken it down to a little extent, but not totally, because the rest of the digestion, if the digestion is supposed to take place at all, is supposed to happen in the small intestine, right? Substrate for fatty acid and glycerol, of course, is the lipid, not the fat. Better than that, you've got the lipase for that. Trypsin is supposed to go for protein, or you could write peptide as well. So you can write protein here, or you can write peptide, which of course will eventually convert this into amino acid again. And you've got the enzyme for glucose. So substrate is supposed to be maltose that breaks down to glucose, and the enzyme is what breaks down maltose, and that's maltase. So that's basically what it is. Now let's go very quickly to the name of the structure that increases the surface area for absorption in the small intestine. Everybody knows that. You can either call it villi and microvilli. So that was a straightforward question. Let's go to some questions that have to be addressed. So one question that was supposed to be addressed is the pyramids. Sorry, the numbers, that is. So we were talking about pyramid of numbers that you need to know. I hope this is what was being asked, versus pyramid of the biomass, right? And in that sense, what is being asked here, that numbers basically represent what? So that each bar represents, or the length of rather, each bar represents the number of that particular organism, Okay. Whereas the width, the length of each bar in period of biomass represents the biomass, or the weight in kilograms, if you will. Mind you, not necessarily the one organism, but collectively in all the organism, I would say. Abdullah Kashif, maltose does not break down to sucrose because maltose itself is made up of glucose and glucose. Maltose is made up of 2 glucose units. Sucrose basically is made up itself of glucose and fructose. So please revise that. Okay. So I'll get back to that in you. मुसद्दिक polypeptides भी सही होगा, peptides भी सही होगा. Polypeptide is often interchangeably used with protein as well. So peptide एक बेहतर लफ्ज़ है इस्तेमाल करने का, polypeptides भी किया जा सकता है. खैर, let's get back to the point. Number of that particular organism, and then obviously pyramid of biomass is the weight in kilogram. So what does that mean? So we'll have to take an example here. And the idea for me is always remember that length of pyramid, generally if it's upright, that's in a perfect scenario. Upright pyramid का मतलब है कि शक्ल ये होनी चाहिए कि, for example, lower bar, ऊपर वाली bar, फिर ऊपर वाली bar, फिर ऊपर वाली bar, right? That's what an upright pyramid kind of looks like. And when will that happen? That will happen when you have a normal scenario there. For example, the food chain that you have is grass, providing energy to caterpillar, providing energy, let's say, to small bird, provide energy to big bird, whatever the big bird is. Okay? So it is obviously making sense that सबसे ज़्यादा grass होगी इस area में. This is something that you have to assume. You cannot always say that, sir, we haven't been given the number. What if घास कम है? What if caterpillars ज़्यादा थी, birds कम थी, and so on and so forth. So don't go for that. Grass is going to be at the edge, and this is going to be the producer, also known as the, that. And then we've got the next one, which is caterpillars. So obviously lots of caterpillars around. Then you've got small birds, which will be slightly lesser, and big birds will be even greater. Why is that the case? Why am I so confidently writing it in this way? It's because I know that in a stable dynamic equilibrium or a stable ecosystem, the number of producers should always exceed everything else. Similarly, the smaller the size of the organism, the greater they have to be in number. Because if the smaller their size they are, and also the smaller in number they are, that means their immediate consumers, which are these three, will struggle. There will be competition for food. So to replenish the energy that they need, the nutrients that they need, it's important to remember that that is what, supposed to be like. Now, this could go a little haywire. So for example, it could even be an inverted pyramid. Now, what does an inverted pyramid mean? Inverted pyramid का necessarily मतलब ये नहीं होता कि पूरा का पूरा pyramid उल्टा हो गया. ठीक है, इसका सिंपली मतलब ये होता है कि आपका फॉर एग्जांपल आपका अगर जो था let's say if I kind of change it around, but let's say that your food chain was tree, caterpillar, once again, same thing, small bird and then big bird. Okay, then obviously अब tree तो एक ही है, so वो एक छोटा सा आएगा. Then caterpillar ढेर सारे हैं probably उसके ऊपर. फिर small birds, I mean trees से ज़्यादा हैं मगर caterpillar से कम होंगे. फिर big birds ज़ाहिर है, caterpillar, small birds सबसे कम होंगे. So now this is looking like an inverted pyramid. This is an inverted pyramid of numbers. And tell you what, more than 99% of the time, if they ask you to make an inverted pyramid of numbers, or they already show you an inverted pyramid of numbers, and they instead now say that please make a pyramid of biomass from this, understand that 99.99% of the time, it is most likely that when you convert this pyramid of numbers right here to pyramid of biomass, it is most likely going to lead to an upright pyramid. It is most likely going to lead to an upright pyramid. And that will solve the mystery, because once again, if I just give you this pyramid and I'm like, okay, you got a tree, caterpillar, small bird, big bird, give me the pyramid of biomass, and you'll be like, okay, tree सबसे ज़्यादा है, मैंने बड़ा बना दिया. Caterpillars सर कितने हैं, small birds क्या ज़्यादा हैं, कितना mass है, कितना कम है, सर एक small bird का mass का तो कम होगा, big bird से, big bird का तो mass ज़्यादा होगा. So all of these arguments will come into play. So that means what you're basically looking for is you're looking for the fact that, okay, once again, tree has the largest, so upright होगा इसका, ठीक है. Then जितने भी caterpillars हैं, काफी ढेर सारे हैं collectively, so उसका उससे कम. जितने भी small birds हैं, ठीक है, उनका weight collectively उन caterpillar से कम, and big bird hardly एक या दो रहेंगी, so उसका उससे कम. So 99% of the time, if they ask you to make a pyramid of biomass, this is what they're doing. This is the reason why we make pyramids of biomass as well as the pyramids of numbers. ठीक है. In some rare scenarios, the pyramid of biomass can even be inverted. In a rare scenario, there could be inverted pyramid of biomass as well. Now, what does that mean? That means that, for example, you've got inside of the water, you've got phytoplankton, which is being consumed by zooplankton, which is then being consumed further by, let's say, small fish, and then, of course, large fish, so on and so forth. So phytoplankton are basically plant-like microscopic organisms. So think of them like plant-like microscopic organisms. Zooplankton are animal-like small microscopic organisms. In other words, basically, they're both microscopic organisms as they are. They're microscopic in their nature. So the thing about microscopic organisms is that their mass keeps on increasing and decreasing based on the number. So there is a chance that the phytoplankton ka mass thoda kam ho jaye, thodi der ke liye zooplankton ka mass zyada ho jaye, and then obviously large fish and small fish. So for a very period of time, the pyramid of biomass could be inverted. But this is for a particular period of time. So this is a rare case scenario. Okay? And yeah, so that's basically it. Musaddiq, if the question is pyramid of energy, well, yeah, I mean, yes, in the case that that energy decreases along a food chain. So for example, if the energy comes to the phytoplankton, we know that about 2% of the energy coming from the sun comes to the phytoplankton. So for example, agar jo energy, ab example hi hai, let's say अब कई ज़्यादा होती है से मगर 200 kilojoules अगर आ रही है phytoplankton से, so 200 kilojoules का जो भी 2% होगा वो phytoplankton को आएगा, and whatever goes from the phytoplankton to zooplankton, that'll be only 10% of that. And then finally to small fish, only 10% of that. So 90% will be lost along the food chain. Remember that energy flow is linear and nutrient flow is cyclical. Nutrient flow is cyclical. Okay? So that's basically the case. Ruby, what is ileum? Ileum is the lower part of the small intestine, which is the curved part. So after the stomach, you have the coil part and the not so coil part. So the upper part is called the duodenum. And this portion over here is called the ileum. So that's the lower part of the small intestine. The ones that got the villi and microvilli, so on and so forth. So yeah, so I think the kids are doing a good job with answering that question. So no worries there. What is more suitable? I think the more specific you are, the better. So ileum is always preferable to duodenum or ileum is more preferable to the small intestine. But again, it differs. So for example, agar aap baat kar rahe ho digestion of fats ki, right? So digestion of fats duodenum mein begin hoti hai, ileum mein end hoti hai. So again, there are two different parts of the small intestine that we're looking at. And if you're asking about digestion of maltose, then that's not happening in the duodenum because pehle starch ne maltose mein break down hona hai duodenum mein. और फिर maltose ने glucose में break down होना है ileum में, इतना time लग जाएगा. So again, there can be two different things happening in both areas. Okay, yes, this is actually an important thing that somebody's pointed out. Yes, you have to know one thing, guys, and before I go with others, because there's quite a lot of questions coming in, the DNA structure and function is going to be important. And this is obviously a shout out to both 0610 and to 5090. And in that sense, you have to build on your information coming from biological molecules chapter, which is what a DNA RNA is made of, all the way till the chapter of inheritance or, for genetic modification. So DNA structure and function may please remember that the monomer, if you will, or which is also known as the simplest unit, represented by what? It is represented by a nucleotide, right? So simplest unit is basically the nucleotide. And a nucleotide is not just the base. The nucleotide consists of three things. One, it's got to have the sugar. Two, it's got to have the phosphate group. And three, it's got to have a nitrogenous organic base, which could either be an A or a T or a G or a C, right? So this is one nucleotide. And another nucleotide will be made in what way? So there's going to be phosphate group coming in here and another sugar. So I'm going to call this a deoxyribose if it's a DNA that we're talking about. So deoxyribose sugar, and of course, another of that. And then, of course, I'm making a third one. So this is one of the strands. The other strand will be met, for example, I'm making the yellow one here in this manner that you've got the organic base first, you've got the upside down sugar, and you have, of course, the phosphate group, and then you have another upside down sugar, and you have another, that's basically how you're creating and you're making up the DNA. And these particular bonds between the sugar and the phosphate, which represents the sugar phosphate backbone, by the way, are basically called the phosphodiester bond. So this bond is called the phosphodiester bond. Whereas this stuff over here, and obviously the bonds between the organic bases, which are these and these, are the hydrogen bonds. They are the hydrogen bonds. Take it? So that's how basically whole strands are made. Now, the important thing is to talk about their function. So obviously DNA carries the instructions, right? Which basically means it carries the genes coding for a particular proteins, right? So they carry the instructions. Now, it follows that two things are going to happen as a result. There's going to be one concept called transcription, in case the 5090 people were not aware of this. And there's the other one called the translation. Now, I'm not going to go into detail, so I'm going to really go quickly over this. I might be going a bit, in a way, okay, I'm going to mix things up, and I'm doing that deliberately because tell you what, this is the first time that you all will be tested on the new syllabus. The last thing you want is not to go through a particular part of the syllabus that you thought might be part, but you didn't go for it. So that's why, sorry for that, but I have to talk about this. Okay, transcription, translation, kya hai? So transcription is basically, in a nutshell, what? Isme kya hota hai ke the DNA double-stranded, obviously it's a double-stranded helix, that separates, okay? One of the strands ends up becoming what we call the template strand, which means what? Which means this is the template strand in black, and connected to the template strand will be different organic bases. So for example, if there's an A here, the T will come here. If there's a G here, the C will come here. If there's a C here, the G will come here, so on and so forth. But I'll just be a bit careful here. I'm not going to mark a T here because what I'm making essentially is not an DNA. This blue thing that I'm making, तो ये G के साथ एक C आ गया, एक C के साथ एक G आ गया, एक और T के साथ A आ गया. This blue thing that I'm making along is not the DNA. So this is the template DNA strand. I'm using that as a template to copy. और ये है RNA. More specifically, इसको mRNA कह देते हैं, जिसको messenger RNA कह देते हैं. So messenger RNA क्या किया? This is transcription. Messenger RNA basically copied the code, DNA code. Copied the DNA code it did. Okay? And once it does that, the messenger RNA exits the nucleus. It's running away because now it has the code. So it exits the nucleus. The messenger RNA is now there. So this is the messenger RNA. And now it needs to know what to do, how to make the protein. So what it needs to do is translation. It does not understand the code. It does not understand the code. In comes to help is a ribosome. So that's what ribosomes do when we say that they're involved in protein synthesis. So this mRNA happily goes to the ribosome and attaches itself to the ribosome. And on top of the ribosome, there's another RNA, which of course is not important here. So the amino acids will start forming on top of it. So for example, here, one polypeptide will basically, amino acid will come in the sense how. So for example, if your mRNA reads ATG, so that means your amino acid will read TAC. And a triplet of this nucleotide sequence, if you will, converts into one single amino acid. So for example, once you're making this, and let's say it's GCT, this CAG, so on and so forth. So you're basically creating an amino acid. So what is happening in transcription? You're copying the DNA code. And what is the mRNA doing after that? It is translating that DNA code into amino acids. So for example, if this was the mRNA, which had, let's say, A, T, sorry, it doesn't have a T, keep saying T here. So it is U, G, and then C, then G, then U, then A, for example. So what will the, and this is 1, 2, 3, 4, 5, 6, 7, 8, and 9. So let's say there's a 3. So 3 of these will have the opposite. So that'll be U, A, and C. Then 3 of these will be the opposite again. That'll be G, C, and A. Then A, U, G ka opposite again ho gaya. U, A, and C. So this represents one amino acid. This represents second amino acid. And this represents third amino acid. So by virtue of this, you've managed to translate the mRNA code, the transcripted code, into 3 amino acids. Into 3 amino acids. So in a nutshell, what's happening? Transcription is the copying. The DNA template strand was created because you had the DNA separate. So the template strand is there. And the template strand से क्या बना? Messenger RNA, उसने copy कर लिया code, उसको code समझ आ गई है. अब वो निकला है बाहर, it does not know how to make the protein, so it employs the use of the ribosome. Employs to use the ribosome. Obviously, details will be done in AS. And the ribosome will then help in making amino acid. Remember that each sequence of 3 nucleotides makes 1 amino acid. So that's why the whole polypeptide is made. And of course, that obviously can be made anew. So that's how you get an enzyme, or you get an antibody, or you get your hair, your nails, so on and so forth, whatever it is that you get. Okay? So that's basically what that was all about. Okay, so Abdullah, this is just one aspect of it, and the others that I wanted to discuss. Mohammed, maybe you might need to join for an A-level session. That's probably going to be more relevant to you. Wa alaikum salam. Nice to see you there. Josefa, is there anything else you want to ask? Okay, let's go above a little bit, because I thought that there was a few more questions coming in. The last 5 minutes to go. I'm just going to basically take the last question now, guys. If there is any specifically. Yes, upright hota hai, peptone, ripsin. Musaddiq, ripsin ka mention kiya ja sakta hai, but for me, in the marketing scheme, etc., usually trypsin and pepsin are mostly written. Pepsin in the stomach, trypsin in the small intestine. If you want to talk about a ripsin, you can. There's no problem about it. But then again, you know, it's up to you, just to make sure that you don't mistake. Okay, there's someone called Sanitizer, whatever that means. What could be tested most likely? I'll tell you at the end. What is what's happening? There is codominance. Okay. All right. We got the structure of the nephron. Okay, so for 5090 people, structure of nephron is really important as well. You've got what we have is the Bowman's capsule, which then merges into a convoluted tubule, which then goes down all the way here, and then here, and then we've got something called the collecting ducts. So without making it too complicated, because it already is, as you say, this portion, if the nephron falls, the above portion, this above portion usually is where the cortex is, which is the outer part of the kidney. There are millions of these, and if it falls over here, then it's the medulla, which is the inner part of the kidney, of course. And then what do we have? You've got you have this particular arteriole coming in. So it depends where you're coming from. So let's say this is where it's coming from. So this is going to be the afferent arteriole, the blood going in. And the one that's coming out is the efferent arteriole, right? And obviously, what do we have in the middle right here? This is the glomerular capillaries, otherwise the glomerulus. It is basically a network of capillaries, right? And this scenario over here is the proximal convoluted tubule. Take in the loop of Henle, then you have right here the ascending and descending limb, and finally you've got the distal convoluted tubule, and this is the collecting duct. So the main thing that happens, number one, in this side, which I'm basically over here, just going to rub this portion off right here. So yeah, this portion right here, gentlemen and ladies, is where the ultrafiltration takes place. Please note this ultrafiltration mechanism happens not just here, but also when it comes to formation of tissue fluid right after any arteriole in the capillary surrounding any random organ. Okay, so ultrafiltration is not diffusion, it's not active transport, it is the filtration of all molecules that are small enough to cross through the capillaries. Remember that capillary cells on their own do have gaps in between. Those gaps are, whatever they are. You can know that. Okay, and basically all the useful stuff actually gets out into the Bowman's capsule. This is the Bowman's capsule for you, and the Bowman's capsule, of course, is part of the nephron itself. So the Bowman's capsule will eventually end up having what? It will end up taking the glucose, which we don't want to lose. It will take all the mineral ions, some of which we do want to lose. It will take in all the water, mostly, some of which we do. It will take all the urea, which we don't want. It will take the amino acids, which we do need back, so on and so forth. However, from the proximal convoluted tubule onwards, which is number 2, then what starts to happen? The concept of selective reabsorption begins. What does that mean? That means that some of it we want back. So we want all the glucose back. We don't want to lose it. If we lose glucose from here, that means we probably have diabetes, which is the failure of insulin to convert glucose to glycogen. Then we've got amino acids as well. So that's what we want out. We don't want all of it in. We don't want any of the urea to come back, but some of it might come back. So a little bit of urea might come back, considering. But basically that's about it. Some of the water might come back, some of the mineral ions will. So again, some of these things will happen. Now, obviously, loop of Henle, etc., will always involve the action of water coming back in. And by the way, this is the area, by the way, where the hormone ADH, which is secreted by the pituitary gland, travels in. And what the ADH does is basically it allows more absorption of water back in to the body, because what it does, it provides more proteins. And of course, that when your body is filled with water and needs to lose water, you need to urinate more, then lesser ADH is released by the pituitary gland. So this is basically in a nutshell what that is about. Okay, that's fine. And righto, so last few points left. The question as to what will come in the exam. So here's the thing, if you're talking about 0610, again, I have no idea what the paper is. Let's be honest, nobody does. But the best we can do is actually make sense of it and try to understand how much, we can do that. Obviously, it depends on what the previous papers had. So I didn't obviously get the chance to do the whole paper. I can, if you want, send the mark scheme in case people don't have the 0610 P4 mark scheme for March 2023. In case I'm not in touch with you directly, you can contact Ahmed Saad to contact me regarding that matter. So for me, for 0610, it's kind of a big goal, but for me, I do believe biotech and genetic modification is the future. So for me, the questions related to them will actually be important. The questions related to conservation will also start being important as well. Something that I discussed in the beginning. For 5090 specifically, I do believe fish stocking will be important because I think that's basically what's going to be answered, especially towards the end of the scenario. And the structure of the nephron, by the way, something that is coming to 5090 for the first time. So the thing that we just discussed about the functions of the nephron is also probably on the cards, because these are things which are reintroduced into the syllabus for 5090. And for 5090 specifically, I'm guessing they're happening. Fish stocking is for both conservation, biotech, and genetic modification, obviously for both, right? So that's basically what I think is going to happen. Yeah, so that's basically it. And yeah, small b, big things, b, big things. I don't know those. Okay, who else? What else? Yeah, vital capacity again is simply the maximum volume of air inhaled followed by the maximum volume that it is exhaled. That's basically what it is, right? And of course, remember that as a result of exercise, your breathing depth, the volume, as well as the breathing frequency, both increases. In aqueous membrane, you're asking about fish stocking. Basically, it's conservation of fish. So we talked about it right there in the beginning of this particular lecture. So if you can probably hope that this will stay online, you can actually take a look at it once we're done with that. And that's basically what it is. And by the way, everybody's asking about fish stocking. Basically, what I mean is conservation. So conservatories of the fish. And how that basically is going to work. ठीक है. फॉर बायोटेक जेनेटिक मॉडिफिकेशन का मेरे से पूछते हैं भाई बायोटेक में क्या था यार. बायोटेक में बेसिकली जो इम्पोर्टेंट थी वो थी पेनिसिलिन की प्रोडक्शन. बिकॉज अब याद रखें पेनिसिलिन की प्रोडक्शन के वो कंडीशंस मांगेंगे. सो उसके लिए पीएच टेम्परेचर खास चाहिए. ठीक है. उसके लिए आपको यू नीड न्यूट्रिएंट्स एंड न्यूट्रिएंट्स के खास पता होने चाहिए आपको. फॉर एग्जांपल ग्लूकोज आपको चाहिए फॉर एनर्जी, obviously for aerobic respiration. आपको ऑक्सीजन चाहिए once again for aerobic respiration. आपको अमोनिया हो सकता है चाहिए हो because that ammonia can be used by the microorganisms to grow and can be used as a source of nitrogen. By the way, ये किसके growth के लिए? This is of course when you place a starter culture of what? You place a starter culture of penicillium. Penicillium is it's the name of the fungus that is supposed to cause production of penicillin. So obviously ये किस पे रखते हैं हम? Fermenter में रखते हैं. And you, after a little while, you realize that the penicillin starts to grow. So for example, if this is a graph and this is time and this is, so the blue line represents penicillin start to grow and then it goes down, and the red line represents that the penicillium is starting to grow, because obviously penicillium is something that is excreted. This is penicillin, and penicillin rather is something that is excreted from the penicillium. So antibiotic की बनाना आ सकता है आपको. ठीक है. That's one thing. Then there's another one. Mycoprotein is something that I'm expecting in there as well. That's also part of the syllabus. That's basically how protein is made by the fungus. This is, by the way, both for 5090 and for 0610 as well. So that's what I'm expecting as well. What else? We got genetic modification, basically how insulin is produced by using plasmids. Okay. So that particular process, you can take a look at that as well. That's in there too. Okay. Righto, so that's time. I need you guys to now concentrate on the following. So yeah, एक तो ये biotech genetic modification है, conservation की बात कर चुके हैं, function nephron. Again, this is all what my basic information is, and I think that that's how you should go with it. 5090 वाले classification के chapter को please देख लीजिएगा, especially the characteristics of arthropods and megapods. IGCSE, I'm not reiterating a lot for you guys because again, आपके already उस syllabus के अंदर है. And for IGCSE, I told you, okay, go for biotech as well. Population control का sigmoid population जो है curve को जरूर देख लीजिएगा. Obviously, सारी चीजें इनमें से कोई ऐसी चीज नहीं है जिसको मैं कह सकूं कि यार, again, it's not going to be asked. What if it does? But then again, we never know what will be. So this is my guess. Could be whatever it is. Guys, please listen. Both of you, 5090 and 0610 people, will be appearing in the morning AM. I understand that some of you have a math paper as well, but the idea is that you guys please make sure that whatever you study, उसको end पे किताबें बंद कर लीजिएगा जो पाकिस्तान में बैठे हैं इस वक्त. ठीक है, for especially for those, and take at least a minimum. Taha, you're right, sigmoid is an IGCSE, but it's also present in the new O levels book as well. Taha, usko zarur dekh lena sigmoid ka. So please make sure that you have at least 7 to 8 hours of sleep minimum before you come. Okay, it's important, and especially in a day that we're having 2 CIs at the same time, it's important that you guys are well rested and fresh when you come in the morning. ठीक है? मुझे नहीं पता किसी का अगर है तो मैं उसकी बात कर रहा हूँ. But anyhow, point is कि in any case आपको 7-8 घंटे की जरूरत है पेपर के. ठीक है? So anyhow, please make sure that happens, and I wish you the best of luck. And yeah, so hit me up if you want, and see you probably before the next lesson. Thank you very much. Assalamu alaikum.
T minus 3.
थैंक यू सो मच सर, बहुत शुक्रिया आपका। एंड आई थिंक यू हैव इंश्योर्ड कि ये जो बच्चे हैं इनका डेफिनेटली इंशाल्लाह स्टार आएगा, और हमारी सबकी दुआएं आप लोगों के साथ हैं। अगेन सर का बहुत बहुत शुक्रिया कि उन्होंने टाइम निकाला। एंड या ऑल द बेस्ट गाइस, अल्लाह ताला आप लोग आपको कामयाब करे। टेक केयर, अल्लाह हाफिज।